Good afternoon, everyone, and welcome to the final presentation of Research Spring 17. My name is Rebecca Traherne. I'm a master's student here from the University of Exeter, and under the supervision of Dr. Travis Van Leeuwen, I am researching the effect of lower salinity on lionfish, including their growth, metabolic phenotype, and consumption rates.
Good morning. My name is Cameron Ergoose. I am the research fellow with the Island School, and together with Becky, we have had the privilege of advising this semester's Lionfish and Mangroves team. So without further ado, pass it over to the team.
My name is Okyle Kislasi.
I'm Ainsley Green. I'm Nina Sanchez.
I'm Andrew Treat. I'm David Mohammed. I'm Nevin Ketchum.
And we're the Lionfish and Mangroves team studying the interactions between schoolmaster snapper and the invasive lionfish. So, as the other group discussed, lionfish are not only an introduced species, but a fairly invasive one, showing the ability to survive in a series of pretty rough conditions. What we know about salinity levels is that they can survive in salinity levels as low as 7 parts per thousand, which is basically fresh water. And so this is part of the reason why we're focusing on lionfish and mangroves. An independent study done by our researcher Becky studied the growth rate of lionfish in 3 different salinity conditions. One is 10 parts per thousand, which is again is almost fresh, basically fresh water, and it showed that the growth rate was negative. The control group, which is a sea-like, ocean-like salinity level, which is about 36 to 40 parts per thousand, is blue here, and it's fairly neutral. And then the 20 parts per thousand, the lionfish in the 20 parts per thousand tank show that they not only stayed about the same, but actually grew, showing that lionfish not only survive but thrive in the mangrove ecosystems. Why we're studying lionfish in mangroves, another reason is there have been 2 studies that showed and basically mapped out not only that lionfish live in the mangrove ecosystems, but they basically said, more research needs to be done because mangroves are pretty important. Not only for humans, we produce a lot of runoff. Basically, the mangrove's kind of the filter between land and sea. Our waste goes into the water, the mangrove kind of filters it before it reaches the ocean. Conversely, the mangrove actually helps creates and produces a lot of oxygen. It's kind of a balance. But more specifically for our study, we're going to be focusing on the ecosystem benefits or the habitat benefits of mangroves. So mangroves provide a sanctuary for juvenile fish. They're a very good nursery. I'll get into that in a second. It's a safe breeding ground for fish to come, mate, lay their eggs, and there's abundant food supplies. The way the mangroves ecosystem works is during high tide, not only are the nutrients flushed in, but so are the predators. And this means that the juvenile fish they need to retreat into the mangrove systems. They're able to do that. And then at low tide, when the predators swim back out of the ecosystem or back out of the mangrove, the juvenile fish are able to come out, explore, and collect some of that nutrients and basically feed.
Past research has shown that lionfish have caused other reef fish, such as the grouper, to change their behavior. And an example of that would be them going out of their reef structure. If this happens in a mangrove ecosystem, it could cause a lot of problems. In the past, lionfish have caused reef fish population to decline, and because the lionfish are such ferocious predators, and in the mangrove ecosystems there's a lot of juvenile fish, this could potentially happen in the mangrove ecosystem. Our study question is, do lionfish cause dispersal of schoolmaster snapper in a mangrove ecosystem? We predicted that the schoolmaster snapper would go out into the flats where they are at risk of predation. We plan to study the interactions between the schoolmaster snapper and the lionfish, how they interact with the mangroves, and the impact of the lionfish in the mangrove ecosystem. To collect the lionfish, we took boats out onto patch reefs and sent in snorkelers, and if there was a presence of lionfish on the patch reefs, we would send in people, and using scuba and hand nets collected the lionfish. To collect the schoolmaster snapper, we went to Page Creek, and by using seine nets and metal traps, collected the schoolmaster snapper. As you can see, each of these different creeks, we took preliminary transects test. to see what types of fish were in the areas that we were studying. In this red circle are the Patries, where we collected the lionfish, and next to it, Page Creek, is where we collected the schoolmaster snapper.
For our experiment, we used juvenile or small schoolmaster snappers. We chose juvenile schoolmaster snappers because they are commonly known to inhabit mangrove ecosystems. However, we varied the size of the lionfish on a continuous scale. We did this because it's not known whether lionfish inhabit mangrove ecosystems, specifically as juveniles or adults. Our experiment was conducted in the wet lab at CEI. As you can see here, this is our troph tank. Located on one side of the troph tank, is the mangrove, which served as the shelter for our experiment. We also used a plastic divider to separate the individuals during their acclimation period. We used video analysis, so our GoPro was located on this pole and filmed from above. This diagram represents our experimental setup. As you can see, the lionfish and schoolmaster snapper are separated by this black line, which represents the divider. The schoolmaster snapper and red lionfish were allowed to acclimatize separately for a total of 5 hours. After this acclimation period, the schoolmaster snapper was filmed by itself for an hour and a half. Then, the divider was removed and the schoolmaster snapper and red lionfish were filmed together. This happened for an hour and a half and between the times of 4:30 and 6 o'clock. We chose this time because red lionfish are known to be crepuscular, which means they are most active during dusk and dawn. After this, the divider was then put back up, the red lionfish was moved to the side of the tank with the mangrove, and then we filmed just the red lionfish for an hour and a half. We repeated this for a total of 7 trials. We utilized quadrats to determine the position of the individuals throughout our experiment. As you can see, on the bottom we have letters A through J. If the lionfish or schoolmaster snapper were in columns H or I, they were considered to be in the mangrove and using it as a shelter. As an example, the red lionfish is in column E and row 2. So we would record the position of this red lionfish as E2 and out of the mangrove. Our data was collected through video analysis. We did this by stopping the video every 2 minutes and recording the position of the individual.
So very recently, we collected results from our experimental trials. As Nina mentioned, during the trials, we were using video analysis, and during this analysis, we would mark the lionfish and the snapper as either in or out of the mangrove. Now, from this data, we calculated the percent occurrence of both the lionfish and the snapper for the snapper solo, the lionfish solo, and the snapper and lionfish interaction. Now, from these percent occurrences, we conducted a chi-squared test. Now, what this test basically does is that it takes what we predicted to see in the solo trial to what we actually saw in the interaction trial between the lionfish and the snapper. And from this test, we found that the snapper had a p-value of less than 0.01. Now, what this indicates is that between the solo interaction trial, the snapper use of the mangrove changed dramatically. As you can see, during the solo trial, the snapper had a very high percent occurrence in the mangrove. However, during the interaction trial with the lionfish, it reduced very much so, which was very key to our findings. The lionfish, however, had a p-value of about 0.7. Indicating no real change in how the lionfish used the mangrove during that interaction trial. And you can see it visually too, between the solo interaction trial. We included this last bar over here, showing the percent occurrence of both the lionfish and the snapper in the mangrove at the same time. And as you can see, it was very rare to see them both sharing the mangrove during our experiment.
So what Andrew and Nina just presented to you was the process of how we gathered our scientific data and the actual scientific data that we gathered. And in more simple terms, what it means is that we actually found that the schoolmaster snapper's behavioral patterns changed while in the trough tank with the lionfish. And while this is so, the lionfish's behavioral patterns did not change. And what does this mean even further, as Ainsley stated earlier on with our hypothesis, is that we expect to see now that the lionfish are invading mangrove ecosystems, that it will be causing the dispersal of the schoolmaster snapper out of the ecosystem and into the open, where they will be at a higher risk of predation. It's due to less shelter.
Wider implications our findings may have on marine ecosystems around us are that if the juvenile snapper populations in the mangroves do decrease, they will no longer be able to reach maturity and make their way out to the reefs. So the reef population of snapper could decrease. Furthermore, snapper are commercially valuable fish species, so it could have major effects on our fishing industries. Further research that could be done in this field are long-term behavioral impacts of the lionfish and the snapper in the tanks together. We actually never witnessed the lionfish feed on the snapper. All we saw were behavioral and territorial aggressions from the lionfish to the snapper. And so the snapper could realize that they are in no physical harm from the lionfish and make their way back into the shelter. Another research study could be the impacts on other species, because other species could use the mangrove for different reasons. This is our literature cited. Our acknowledgements. We'd like to send a huge thanks to Cameron Raguse and Rebecca Traherne, the Island School, Cape Luther Institute, and the University of Exeter. Are there any questions? Yes.
So lionfish are, I believe lionfish are generally territorial. So I guess my question is, would they prefer to be more in a reef setting, or are we finding that they're just exactly in the mangrove, just kind of where they live? Are they being born in the reef and then moving into the mangrove, or vice versa?
Good question. So I believe you're asking about do lionfish prefer, where do they prefer to live? Is it the structure? I would say my opinion is that lionfish are known to like and prefer to live within a structure. A lot of times when you go dive, when we go looking for them, we have to actually go look inside of the rocks, inside of the corals. The reason why we're studying lionfish in mangroves, and maybe this is something that could be potentially researched, is we don't really know if lionfish are living in the mangrove system as juveniles and then moving out to the reefs. We don't really know. That's why we vary the size, so that error wouldn't potentially be an error for our data. But I would say that it doesn't really matter. Lionfish actually, Becky's research showed that they do better in lower salinities like the mangrove system. So from what we've seen and the research is out there, it's just they prefer structure, whether it's a mangrove or a reef, a coral reef. We don't really know where they prefer to live in terms of the larger ecosystems.
I found the map fascinating to watch the lionfish growth over the years. And I'm wondering, since they are comfortable in low salinated water, how much have gone upstream already into the U.S. and have they seen that pattern?
So the question is, because they're comfortable living in low salinity, have they been going upstream to the U.S.? There have been reported sightings of lionfish as far up as Maine. I'm personally from New York, and we know that they're in the Hudson, which is pretty brackish water. So my answer would be yes. And at the reproduction rate, I believe that they have the potential to continue moving upstream.
Great Lakes.
Yeah. I mean, I don't know. That's very fresh water, but who knows?